Okay, we're talking today about the creation of protein synthesis. This is one of the coolest functions that go on in cells. This is, for me, one of those evidences that God exists. We talked when we were just introducing you to biomolecules back in chapter 2. We talked about how proteins need DNA to be made, and DNA needs proteins to be made. We're talking this week about the first one of those, that proteins are made off of the information from DNA. And the fact that it is so complicated and that proteins are so immense and so exacting just screams that there was an intelligence behind this. You can't get a process this complicated with so much information in it by chance. Explosions do not create information. Random events do not create information. And so the process of a protein being made is a reason to worship. And so as we are looking at this process today, there's a couple of big steps, and I want to cover those in overview in this first slide, and then we're going to look at more detail on how all of these things happen. Okay, so DNA is the information repository. It's the library of your cell. You can think of it that way. It's where all the information for what the cell is going to do resides. And that information is stored in the nucleus. So DNA is a language, and it's written in 4 letters. We've covered this before. And each word is 3 letters long. A codon is 3 letters long. And all of these words, these codons, go together to make sentences that are genes. And all those sentences go together to make an encyclopedia, which is the information for us to make an organism. So all of that DNA is stored and very, very well organized in the nucleus. We need to get the information out of the nucleus, though, into the rest of the cell. Proteins are not made in the nucleus. Proteins are made out in the cytoplasm of the cell. So we can't let the DNA go out of the nucleus because then it would be lost. And it's very important to keep all the DNA in the library. So we don't just send a DNA out into the nucleus to make proteins off of the information that's in the DNA. We make a copy of a gene. And that copy is called messenger RNA. It's made out of RNA, ribonucleic acid, not DNA, deoxyribonucleic acid. We talked briefly about the differences between the two. Ribonucleic acid is only single-stranded, not double-stranded. Ribonucleic acid has uracil, not thymine. So those are the big differences. There's other minor ones. But we make a copy of the DNA into messenger RNA, and the messenger RNA is expendable. Picture, in a library, there is an old book that's very valuable. The library will not let you check that book out and take it home because they don't want it to get damaged and lost. But you can take that book to the photocopier and throw a couple of dimes in the photocopier, and out comes a photocopy of the page or 2 pages that you needed. That's the concept we have here. Messenger RNA is expendable. It doesn't have to be kept. It's used and then recycled. Okay? So messenger RNA, chief copy, made of the DNA, and it leaves the nucleus. It goes to the ribosome. This is an organelle in the cell, in the cytoplasm, that makes proteins. Ribosome itself is actually made up of RNA and protein. So there's RNA that it takes to read the RNA to make protein, and the proteins are part of the ribosome that make the protein. It's very interdependent. You can't have information in the DNA without proteins. You can't have proteins without information in the DNA. They need each other. They had to come to being together. You couldn't have one for certain numbers of thousands of years without the other also being there. So it's a great chicken and egg quandary, and the answer is God. So the ribosome makes the protein, and it does so by bringing amino acids. Remember, amino acids are the building blocks of a protein. It's the monomer of a protein. It brings amino acids to the ribosome, and they are attached to little bits of information on transfer RNA molecules. You'll see pictures of this in a minute. So there's RNA that builds the ribosome itself. There's RNA that is a copy of the DNA message, and there's another kind of RNA that is attached to an amino acid. And it's the mating together of the tRNA and the mRNA that aligns all the amino acids in the right order. And the ribosome is able to join those together and make a protein. You'll see how this works in a moment as we get more into detail. But the complexity here really is astounding. And so, you know, as you're looking at this, just keep in mind that we serve an amazing God. There's two vocabulary words here for the stages of protein synthesis. The first one, the making of messenger RNA, is called transcription. To transcribe means to take information and write it down somewhere else. Okay, kind of like a court reporter. To have information in one form and write it down in another form or in another place. The other vocabulary word for this process, I have nowhere to put my picture now, is translation. Translation is what we call it when we take the information in mRNA and build proteins out of it. So transcription and translation are the two processes that we're going to be talking about. Transcription happens first. Transcription happens in the nucleus. Translation happens in the ribosome, out of the cytoplasm. Okay? All right. There you go. Take a moment, write that down. Okay, so DNA, we said before, it's too important to leave the nucleus, so we have to make a cheap copy. And when I say cheap copy, I don't mean that it's full of errors. I just mean that it's expendable. The cell can build a messenger RNA molecule quickly and use it, and then take it back apart and use the parts to make another messenger RNA. So it's recyclable, it's easily made. If it gets lost, it's no big deal. Okay? So that copy is messenger RNA, mRNA. And just as a reminder, so it's in your notes again, there are 4 nucleotides. There are 4 monomers to nucleic acids, DNA and RNA. Adenine and thymine, guanine and cytosine. And the adenine and the thymine are always paired up on a DNA molecule. When there's adenine on one side of the molecule, there's a thymine on the other side of the molecule. They always occur in a pair. And the same thing is true of guanine and cytosine, where there's a guanine on one side, there's a cytosine on the other. Okay? And so if you have one strand of DNA, you can write the complementary anti-strand to the other side, because they always occur in the same order. So that's true on DNA, and now as we're writing DNA and making a copy of it in RNA, in messenger RNA, there's a little bit of a difference, because RNA, ribonucleic acid, doesn't have thymine, it has uracil instead. So any place you would have written a T, you write a U. Okay? So instead of A's and T's being paired, A's and U's are paired when you're writing messenger RNA. G's and C's don't change. Okay, so in transcription, we get a complementary copy of what the DNA gene sequence was. So let's say that this is a gene, and this would be stupidly short for a gene. Genes are a lot longer than 5 codons long, but just for the sake of example and in your notes, if the DNA said thymine, adenine, cytosine, the complementary messenger RNA would say adenine, uracil, guanine, because T's and A's are always paired. This, if this were DNA, would be an A and another T, because they're paired. But now I'm writing a messenger RNA. RNA doesn't have thymine, it has uracil. So this is a U. G's and C's are always paired. So the complementary, what we could say, anti-codon, the codon on the opposite side of this strand for TAC is AUG. And then you can keep going here. Oh, look at that, there's a mistake. There's a mistake, G's and G's, it would not be GG. It would be GC, I'm sorry, there's an error here. CAU, CAU, And then TCC would become AGG, and GAT would become CUA. And again, the U is here because there would be an A and a T, but I'm writing a messenger RNA, which doesn't have thymine, it has uracil. And then TGC would be ACG. Okay, sorry about that typo, I'll fix that. So it should be CAU, not GAU. Okay, so we have this copy. We have transcribed DNA and made a copy of it in messenger RNA. Okay? The process would look something like this. This is a cartoon, but what's happening is the DNA is unwound in the region where the gene is. And the code on one side is red. The actual gene is only on one side. The other side is just a template strand to help make more DNA in the future. And so it reads one side of the DNA molecule, and it creates the complementary messenger RNA. So again, TAG would become CAU, GTT would become CAA, and as it reads, it creates this messenger RNA strand that comes off the back. And then after it's read, the DNA is allowed to wind back up again. Okay, so you've got to open the book, make a copy, and you close the book up. And the name of the enzyme that does that is RNA polymerase. You don't necessarily need to know that, but that's the name of the enzyme that does this. So again, the interconnectedness between DNA and proteins. It's a protein that reads the DNA to make a copy of the messenger RNA so it can go out and make more protein. How do you get a protein to read DNA if there's not a messenger to make the protein in the first place? It's a cycle, it's a loop. You have to have both at the same time. Okay. There's another picture just of the same thing, just another image of it. So the messenger RNA is created by RNA polymerase, and the DNA is unwound and read, and this molecule will slide along the gene and create RNA, messenger RNA as it goes. Okay. Here's another picture of it. This is meant to look more realistic, but it's still just a graphic. Here's a piece of DNA that's being read by this protein, and it's creating the messenger RNA strand coming out of the back. Huh?
No, it doesn't.
Oh,
wait,
okay.
I see it.
Oh, I thought you were talking about the whole thing all together. I was like,
Okay, there it is. Thank
How
you.
did you see that? I don't know.
Okay, so messenger RNA leaves the nucleus and moves into the cytoplasm, and it gets attached to a ribosome. Now, a ribosome actually has two parts that are made out of both proteins and some more RNA themselves. This interconnectedness keeps going. Ribosomes are they can be free-floating in the cytoplasm, drifting around all by themselves, or they can be stuck to the endoplasmic reticulum. So there are two places you can find ribosomes. Messenger RNA is read 3 bases at a time, and those 3 base words we call codons. So just like in our language, we have an alphabet. You stick letters together to make words, you put words together to make a sentence, and you put sentences together to make a book or an essay or whatever. DNA has the same exact kind of arrangement of complexity. They have an alphabet. Its alphabet is 4 letters big. Its words are 3 letters long. All of the words are 3 letters long. And then those words arranged together make sentences that we would call genes. And those genes, when you put all the genes together, make the volume of the encyclopedia of how to make an organism. So these 3-letter groups are called codons, and you'll see why it's a 3-letter group in a moment. But we read those 3 letters at a time. So in the example that we're working with here, AUG is a codon, GAU is a codon, AGG is a codon, CUA is a codon, ACG is a codon. Okay? And interestingly, every gene starts with AUG in the messenger RNA. That's what we call the start codon. That's what your cell looks for to find the beginning of a sentence. It's kind of like a capital letter. Okay, AUG is the start codon. It's always there, beginning of the gene. Okay, so we have this and I just said that same thing. We have this 3-letter word. If the individual nucleotides are letters in the genetic language, then codons are words, and the whole gene is a sentence. We've already got that. Okay, translation. The ribosome, for the sake of time, I'm going to keep talking. The ribosome attaches to the messenger RNA at the start codon at AUG. It then attaches a complementary piece of tRNA to each codon in the mRNA. So you'll see a picture in a minute. Transfer RNA transfers. It brings things. It brings amino acids to the ribosome. And every 3 letters of the message has a complementary 3 letters of a tRNA. And so they pair up, and they fit together, and they bond. Okay, and then on the back of that tRNA molecule is an amino acid. And so the amino acids are put in the right order because the tRNA sit on the messenger RNA in the right order. And now what the ribosome has to do is attach the amino acids together and release them from the transfer RNA. Okay, so it attaches a complementary piece of transfer RNA to each codon in the messenger RNA. The transfer RNA have amino acids already attached to them. And then the ribosome joins it together. So again, the design of this just blows my gourd. Because not only do we have message in DNA that we found a way to keep precious and safe in the nucleus, but we make these copies that we can throw away. And the tRNA is built by your cell to have the right 3 bases and an amino acid already on it. So these building blocks are just manufactured by your cell. So you've got to have all the machinery that it takes to build the building blocks too. And all of that machinery to build the building blocks is coded for in the DNA. And so you have to have the DNA to make the tools, to make the proteins. It's amazing. Amazing. God is incredible in his design and his ability to create in the smallest details. So the tRNA come and they line up in order, kind of like elementary kids going to lunch. And then the ribosome slides down the message and joins the amino acids together. So you don't do that with elementary kids. That would be kind of bizarre. But picture all the amino acids in the right order, standing there on their messenger RNA saying, okay, here I am. And then the ribosome just comes down and says, you go to put this one, and you join, and you join, and you join. And then cuts them off with the transfer RNA. And you now have amino acids joined together in a polypeptide chain that is in the right order. Okay. I have pictures of this too. Are you guys still ready? Okay. Take a second. So this is a ribosome, and this is the messenger RNA. And so this is a transfer RNA molecule. It has a complementary anticodon that pairs with the messenger RNA. So it's in the right place because the letters line up. And on the back of the transfer RNA is an amino acid. So the next transfer RNA slots in here, and it lines up with its anticodon. Then the ribosome is able to join this amino acid chain to the next amino acid, and then it slides down. And it keeps doing that repetitively. Okay? Here is a little animation of that happening. It's a GIF, yes. So here comes an amino acid, lines up on the mRNA. The ribosome joins the amino acids together on the back. Eventually, you have a long chain of amino acids. After this gets repeated a whole bunch of times. And then at the end of the gene, just like there was a start codon, there's a stop codon. So the amino acids here get joined together. That process repeats a whole bunch. You get a chain of them. And eventually, at the stop codon, there's a piece of tRNA that does not have an amino acid on the back. And so it serves as the end of the gene. The amino acid is released, and the cell continues to process. Okay, so that's what this looks like. This is your last slide. The growing chain of amino acids is the protein. It's a polypeptide chain. Now remember that it's not done when it's just amino acids in the right order. That's the first level of complexity. We talked about the fact that proteins have 4 levels of complexity. This polypeptide chain still needs to be folded into the right shape. It still needs to be put together with other amino acid chains to function in the protein, right? So there's a lot more complexity that has to be done to this. But as far as getting the amino acids in the right order, this is how it happens. When the ribosome gets to the stop codon, there's a tRNA molecule that does not bring amino acid. It's just a dead end. So the ribosome goes to join this polypeptide chain to another amino acid, and there's no amino acid. And so the ribosome releases the protein, and the protein goes off, and the manufacturing of the protein will be continued by the Golgi body and other parts of the cell that are going to add more layers of complexity to this. But we're looking here just at how the protein is synthesized in the ribosome. Okay, so that's the end of the information. Do you guys understand conceptually what happens? Yes? We have a copy, we have precious information in the nucleus, we make expendable copies in the messenger RNA. Messenger RNA has these 3-letter codons. The transfer RNAs line up on their codon. The ribosome moves across the whole line of amino acids, joins the amino acids together, and releases them from the transfer RNA so that they can go off and continue being manufactured. So that's the process of protein synthesis in a nutshell. Okay. Just want to show you some last pictures. I know lots of you are still writing, but I want to make sure I get these pictures for you. If you need to get the notes, the last couple of words, this will be on the blog quickly. So I just want to show you pictures here. So here, this is the whole process. DNA in the nucleus. The messenger RNA is being created there in the nucleus by RNA polymerase. The messenger RNA then slides out of the nucleus and gets read by the ribosome. The transfer RNAs come to the ribosome with the anticodons already, so that they can pair up. The amino acids are a part of that molecule. And the growing polypeptide chain then is going to go and get further processed by the Golgi apparatus. This slide also shows the process of making a transfer RNA molecule, which is beyond what you need to know at the high school level, but is just more and more reasons to worship the Lord with the incredible complexity he creates. So this is another quick little animation, and after this, I will let you go.
In prokaryotic cells, translation is initiated by formation of an initiation complex consisting of the 30S ribosomal subunit, formylmethionine tRNA, and messenger RNA. The 50S ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has 2 sites to which transfer RNA-carrying amino acids can bind. One is called the peptidyl, or P site, and the other is called the acceptor, or A site. There is also a third site called the exit, or E site, where transfer RNAs are released. The initiating transfer RNA, carrying formylmethionine, binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The formylmethionine carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site by a peptide bond. The ribosome now advances a distance of 1 codon, and the transfer RNA that carried the formylmethionine is released at the E site. A transfer RNA carrying the next amino acid now moves into the A site, where the anticodon on the transfer RNA matches the codon on the messenger RNA. The ribosome shifts down by a distance of 1 codon. As the shift occurs, the 2 amino acids on the transfer RNA in the P site are transferred to the new amino acid, and the second transfer RNA is released from the E site. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain.
Does that help you?
Elongation of the polypeptide is...
There you go. We'll deal with this all week. God bless you guys. Okay.